One characteristic of angiosperms is that they all produce flowers and produce seeds that are enclosed in fruits. Hence, they are known as flowering plants. A flower is the reproductive structure of an angiosperm. Angiosperms are the most diverse group of plants. There are approximately 250,000 different types of angiosperms in the world. It is not surprising that they are the most dominant land plant species on Earth, making up around 80% of all living plant species on Earth. In this podcast, we will be discussing the structure of the dicotyledonous animal pollinated flower structure, pollination, fertilization and seed dispersal, the structure of a dicotyledonous seed, germination of a typical seed, and how flowering is controlled in long day and short day plants, including the role of phytochromes. The peduncle is the stalk of the flower. Receptacle, the part of a flower stalk where the parts of the flower are attached. Sepal, the outer parts of the flower, which are often green and leaf-like, that enclose a developing bud. The petal is the part of a flower that are often conspicuously colored. The stamen is the pollen-producing part of the flower, usually with a slender filament supporting the anther. The anther is the part of the stamen where the pollen is produced. Pistil, the ovule-producing part of the flower. The ovary often supports a long style topped by a stigma. The mature ovary is a fruit, and the mature ovule is a seed. The stigma is the part of the pistil where the pollen germinates. And lastly, the ovary is the enlarged basal portion of the pistil where ovules are produced.
The three main processes required for reproduction in angiospermophytes are pollination, fertilization, and seed dispersal. The first process, pollination, is what you see here between the bee and the flower. This process is the transfer of pollen from the anther to the stigma. The structure of the flower is adapted to its method of pollination. The second process required is fertilization. This involves the fusion of the male gamete nuclei in the pollen grain with the female gamete in the ovule, which is located within the ovary. This process forms a zygote, which eventually develops into the seed. Fertilization in flowering plants can only occur once a pollen grain has landed on the stigma and has germinated there. Finally, seed dispersal. This is the carrying of the seed away from the parent plant. Seeds are moved away from the parent plant before germination to avoid competition for resources. There are many seed dispersal mechanisms, namely through fruit, wind, water, and animals.
The embryo is the young multicellular organism before it emerges from the seed. The endosperm is a source of stored food consisting primarily of starches. The seed coat consists of one or more protective layers that encase the seed. The mature embryo consists of an embryonic root known as the radicle, an embryonic shoot known as the plumule, and two cotyledons in this photograph. The hypocotyl is the portion below the point of attachment and is connected to the radicle. The epicotyl is the portion of the embryonic stem above the point at which the stem is attached to the dicotyledon. The cotyledon is described as a seed leaf that stores food in the form of starch and protein for use by the embryo. A dicot stores its food in the two cotyledons.
All these different structural parts of a seed play a role in germination, which is the resumption of growth of an embryo or spore that occurs when a seed or spore sprouts. Essentially, germination is the process in which seeds or sprouts begin to grow. There are many different conditions required for the germination of a typical seed. Firstly, water. Water ensures that the seed is fully hydrated and the embryo is physiologically active. It also stimulates the release of enzymes and washes out any inhibitory enzymes within the seed. Water uptake allows the testa to break and therefore allows germination. Too little water results in the testa not breaking and therefore inhibiting germination, but too much water can also result in the seed becoming oxygen starved and therefore also inhibiting germination. The second condition required for the germination of a seed is oxygen, because it sustains aerobic respiration and generates a continuous supply of metabolic energy in the form of ATP. In this way, oxygen allows for growth, and therefore too little oxygen will inhibit germination. A suitable temperature is also required for the germination of a seed. This temperature should be close to the optimum temperature for the enzymes involved in the immobilization of stored food reserves within the seed. Each species has a unique optimum temperature. As a general rule, an increased temperature leads to increased germination due to increased enzymatic reaction rates, but past the optimum temperature of the enzyme, it experiences denaturation and therefore germination decreases. An energy source is also required for germination. This usually comes from the food molecules stored in the endosperm or cotyledons, which provide ATP to the seed. In particular seeds, water, oxygen, or temperature may need to be preceded by other processes for germination to occur. For example, fire is often required in some plants because it reduces competition and breaks down inhibitors. One example of such a plant is the lodgepole pine, which grows in the chaparral biome and has leaves that are protected with a resin that only fires can melt away. This way, fires allow the release of seeds from the plant. Other processes that may need to precede these optimum conditions are the testa being worn down, possibly by being passed through an animal's digestive tract, as well as the presence of light, such as wheat seeds, which are shown in the picture.
Outline the metabolic processes during germination of a starchy seed. Firstly, the absorption of water, also known as imbibition, activates the cotyledon cells. Water is absorbed by cellulose, pectin, and starches in the seed. The testa often softens or bursts because water causes the seed to swell. Metabolic processes begin to take place. The role of water in the germination of the seed. Water stimulates the secretion of gibberellin, which is a metabolic enzyme. And this in turn stimulates the plant hormone to the thin outer layer of the endosperm called the eleurone. The eleurone makes alpha amylase, which digests the starch of the endosperm to smaller sugars, or maltose. Water also transports the hormones, rehydrates cells and tissues, washes out inhibitory enzymes, and transports the food reserves. The steps of germination. Firstly, the radicle, which is the embryonic root, emerges. The shoot tip begins to develop. The hypocotyl pushes above the ground, and once it reaches the light, it straightens. And lastly, the epicotyl spreads the first foliage leaves.
After seed germination, the growth of a plant is often continued on with the process of flowering, which is different in long-day and short-day plants and is controlled by phytochrome. Short-day plants flower when the day is short, in seasons of spring or autumn. Long-day plants, however, flower when the day is long and the night is short, mainly in summer. So the length of darkness is the main determining factor between short-day and long-day plants. Phytochrome is a blue-green pigment present in green plants in very low concentration. It is a large conjugated protein and it is highly reactive. It is a photoreceptor pigment because it is able to absorb a particular wavelength of light. There are two interconvertible forms of phytochrome, and these convert to one another based on light absorption. PR is a blue pigment which mainly absorbs red light of wavelength 660 nanometers, while PFR is a blue-green pigment which mainly absorbs far-red light of wavelength 730 nanometers. When the PR form, or the inactive form of phytochrome, absorbs red light of wavelength 660 nanometers, it converts into PFR, or the active form of phytochrome. When the PFR form absorbs far red light of wavelength 730 nanometers, it immediately converts back into PR. So how does phytochrome control flowering in short day and long day plants? In short day plants, PFR inhibits flowering. That's why long nights are required so that PFR can convert back into PR, and there are low concentrations of PFR, therefore reducing the inhibition of flowering. In long day plants, however, PFR promotes flowering, or bursts of light can be used to convert PR into PFR. That's why in long day plants, short nights are required so that high concentrations of PFR are present, and thus flowering is promoted. As we have explained in this podcast, there are many conditions that affect the growth and development of a flowering plant, specifically the reproduction of Angiospermophytes. These include both external factors, such as environmental temperatures, as well as internal factors, such as the structure of an angiosperm.